Thanks. So, hi everyone. It's a real pleasure to speak to you today, and thank you for the invitation. I'm going to talk about the evolution of metastasis in colorectal cancer, and I'm going to talk a little bit about the models that we use to make sense of the data that we collect. So this is a talk really driven by wanting to understand metastasis rather than wanting to tell you just about mathematical modeling, but it definitely has some maths in it. And so I'm going to focus on these two very interesting elements of evolution, or perhaps not evolution, if you like, of stabilizing selection, which is definitely evolution, and plasticity, which is perhaps not evolution. And we'll talk about that now. And as you've heard, I'm at Barts and in the Center for Genomics and Computational Biology. So we're interested in understanding how colon tumors progress from benign adenomas through to metastatic cancers. And as many of you will be aware, I'm sure all of you are aware, throughout recent years, colon cancer really has been paradigmatic for understanding the evolution of malignancy. And famously, the work from Bert Vogelstein proposed what is affectionately known as the Vogelgram, where there's a series of key alterations in key cancer genes, which each drive a clonal expansion. And that's what's depicted in this cartoon. We have time on the x-axis versus size of a clone on the y-axis. Each of these red crosses is the acquisition of a key driver alteration, and the expanding waves of color are the clonal expansion of that alteration. And so you can imagine that this is an appealing model to understand metastasis. We might imagine that metastasis is simply at the end of this evolutionary process when a number of these driver alterations have been accrued. And so, of course, we have a good idea of what the key genes are in colon cancer, and we can overlay them here. And so for a number of years, we've really been asking the question, is this true? Is this what really happens? And I want to talk about metastasis, but to give you some foundation for what I want to say in metastasis, I think it's helpful to give you a little whistle stop through our work in understanding the transition from pre-cancers to cancers, and also looking at the evolutionary dynamics in primary tumors. And that will set the stage for the metastasis work. And so, as I suggested, colon really is an exemplar tissue to look at evolution of malignancy in a patient. And it's an exemplar because we have healthy colon, but we also have a very well-defined pre-cancer state of colorectal adenomas that I call CRCs for short. We have colorectal cancers, of course, and then we have metastasis, which is commonly in the liver, but also in other organs such as the lung. And so we're able to access both pre-cancer and cancerous stage relatively easily. And so what we did a couple of years ago was to compare mutational burdens in precancers in these adenomas to cancers. So we compared CRCs to CRCs, precancers to cancers. And this is the burden of somatic nucleotide variants, SNVs, in adenomas versus cancers. And you can see they're roughly the same. There's perhaps slightly more. It wasn't significant in our cohort in colorectal cancers. And so this was a surprise. We have this model that sequential acquisition of mutations underlies progression, but when we compare pre-cancer to cancer, that's not what we see at all. The total burden of SNVs is very similar. And in fact, the burden of driver alterations is also very similar between pre-cancers and cancers in the colon. What is very different genetically, though, is the burden of copy number alterations, so the loss and gain of whole chromosomes or part chromosomes. And here the cancer's on the right, and you can see there's many more copy number alterations. So this is suggestive, obviously not proof, that copy number alterations are causative in colon cancer development. If we then do multi-region sequencing and look at the clonality of these important alterations, so really we're using tumors as a model system here to dig into their evolution. What we find is that the driver alterations are invariably clonal within cancers. So they're in every single cell within these primary colon cancers, and only very rarely do we see subclonal driver alterations in colon cancer. So we did multi-region whole exome or whole genome sequencing, built these phylogenetic trees, these family trees, and annotated on the trees where the drivers are. And you can see a bunch of different cancers here, and you'll see that virtually all driver alterations are clonal. So again, this is a problem for this genetic progression model. This is not what we see when we actually look in the data. And so here's the first bit of maths. We also ask, can we learn about what the evolutionary dynamics are within a primary tumor? And so we wrote down a stochastic branching process model to ask, well, how do the mutations propagate through a tumor as that tumor grows? And within that, we can predict what will happen under neutral evolution So neutral evolution is a situation where all cells within the tumor grow at the same rate as each other. Every cell within the tumor has the same probability of producing surviving offspring. So under neutral evolution, the maths predicts that you'll get a tail of ever more mutations at ever lower frequency. And specifically that shape, that tail of low frequency mutations follows this distribution, and it has a 1/f² form. So there's F here is the frequency of mutations in the tumor. So if you look at that in sequencing data, this is a plot of mutation frequencies from whole genome sequencing. You see this tail of ever more mutations at ever lower frequency in this histogram. And in fact, if you do a mathematical test, you can show that this shape is in fact this 1/f². And so when we look in colon cancers with our mathematical prediction about what tumor heterogeneity should look like if these tumors evolving neutrally, I think to our surprise at the time, we found that the vast majority of colon cancers look like they're evolving neutrally from this pattern of intratumor heterogeneity predicted by the mathematics. So putting that all together then, what does that give us? So we started with this idea of sequential selective sweeps, new driver alterations showing up and driving expansions and that underlying progression. But when we actually look in the data, that's not what we see. And instead we see this situation where it looks like the important genetic action is there at the start. So we have all of our SNVs, we have lots of copy number alterations, and then within that growing tumor, all the evolution looks neutral. So there isn't an expanding clone that shows up later on. In fact, the only thing that determines the size of the clone in the final tumor is when that clone showed up during tumor growth, not selection for it. So we have this idea that all the important evolution, all the selection in a primary colorectal cancer happened at the outset. That first cancer cell is, of course, strongly selected. But then within the growing tumor, all of the subsequent evolution looks neutral. So that's obviously very different to this genetic progression model that we started with. And so how do we reconcile these insights with our understanding of how metastases might develop, or hypotheses about how metastases might develop? Well, so if we look at points 1 and 2, so 1 says the burden of SNVs in primary tumors is the same as precancers, so perhaps SNVs are not so causative in the development of primary tumors. And then in big sequencing cohorts that I'm sure you're well aware of, there aren't a big list of metastasis-specific SNVs that have been found in colorectal cancer metastases in the liver. So perhaps there aren't metastasis-specific driver genes, or at least commonly those genes are not present in metastases. Given our insights in primary tumors, we see that copy numbers do accrue and seem to underlie progression from precancers to cancers. So perhaps copy numbers are really important in metastasis. It's a reasonable hypothesis given our understanding of primaries. And then I think the most interesting one to think about is this observation of neutral evolution in primary tumors. If all cells in the primary tumor are equally likely to evolve, they all have the same probability of producing surviving offspring, how can we get adaptation within a primary tumor? How can there be evolution? Well, there might not be evolution, but there may be plasticity. So if a tumor is neutrally evolving, then any cell has the same probability of adapting to some new microenvironment. We suspect microenvironments to be heterogeneous in primary tumors. So perhaps this observation of neutral evolution points towards inherent plasticity from the outset of colon cancer evolution. Perhaps all those cells in the primary tumor are plastic. And so therefore, perhaps metastasis happens just by random spread and colonization of distant organs through plastic adaptation rather than through clonal selection of a perfectly metastasizing genotype. That would kind of reconcile this lack of a role for genetic evolution that we predict. Okay, so let's go through those. So let's look at 2 and 3, and I'll show you some of our data investigating these. So if we start with 2 and investigate the role of copy number alterations in metastasis in the colon, we found a series of patients working together with Hemant Kocher here at Barts, and more recently with John Bridgewater at UCL, where we had both primary and then sequential metastases. So in this patient, we have a primary tumor removed by surgery, and a year later they had a metastasis. We looked at the pattern of copy number alterations in both primary and metastasis in these tumors. This is a graph of the genome on the x-axis. You have loss and gain of genetic material, gain if the line goes up, loss if it goes down. And if you contrast the pattern of losses and gains in primary versus metastasis, you'll see that they're virtually identical. I find this really surprising. We have a tumor which has moved tissue from the colon to the liver, and it's been growing in the liver for at least a year, probably longer if metastasis happened early. And yet there's no obvious sign of evolution in these copy numbers, even though we think they're causative for the initial development of cancer. This is a pattern we see across patients. So this is a second patient, again, with a primary colon cancer and a secondary liver metastasis, shorter time interval, but this time they had chemotherapy in the interval. Again, the pattern of copy numbers are virtually the same. There are a few differences here highlighted with the arrow, but broadly speaking, there's very little change in the pattern of copy numbers here. It's the same here. This is a really interesting patient. We have multiple metastases in the liver and then later in the lung. They've also had a diverse range of treatments in this time. Again, the pattern of copy numbers don't change very much at all, despite various rounds of treatment and despite going through 3 different tissues here. You can imagine there's a high chance of bottlenecking as cells move from tissue to tissue too. So this, we should see changes in the genomes. So what's going on? Well, one option could be that there is actually no chromosomal instability once cells metastasize. I think that's extremely unlikely. We sequence, this is in primary tumors, I must say, but we sequence individual cells from a single colorectal crypt in primary tumors together with Hugo Snippet in the Netherlands. And these are individual cells selected from one tumor gland here. And again, this is the pattern of copy number alterations with the color code here. And you can see within a single tumor gland, there's quite extensive copy number diversity. So there is ongoing copy number change all the time. But when we look across time and space in tissues, we don't see it. So what's going on? So this is where the maths comes in. So what we postulated was that there could be negative selection. There could be that there is copy number variation generated all the time. Those cells with those new copy number alterations are less fit than cells without those new copy number alterations. And so those newly mutated cells are removed from the population by negative selection. And so we can make a mathematical model of this hypothesis and check that it really does produce the behavior that we expect. So what we do is define some optimal genotype, some optimal carrier type here, defined by this pattern of copy number alterations. It's just arbitrary. And then we have some abstract fitness landscape. It's obviously in multi-dimensions because of all the chromosomes, but we just project it in 2D, so you know what I'm talking about. Where if a cell has the genotype that puts it on the fitness peak, it has optimal fitness, produces surviving offspring. And as it accrues new copy number alterations, it might move away in this abstract genotype space from the fitness peak, be less fit and therefore produce less surviving offspring and be outcompeted, grow slow, or even be killed off in the population. And so we can simulate this model as a stochastic birth-death process, and we define fitness by this value S, which is inversely proportional from the distance of the observed genotype of a cell to this proposed optimum with some constants to make the whole thing scale nicely. So if we simulate that, we can turn up the strength of selection from left to right here in these heat maps and ask how diverse do we expect cells to be within our population. So here, each row in the heat map is the genotype of a single cell in our population. Again, the colors represent copy number alterations. And you can see when there is no negative selection, the population becomes very, very diverse as we let it evolve. But as we introduce stronger and stronger negative selection on those copy numbers, what we see is that homogenizes the population, just as you would intuitively expect. And populations get trapped on this fitness peak. So the only genotype that we tend to observe is the one that is most fit. And so this is suggestive then that the unchanging karyotypes that we observe through metastasis is due to the action of stabilizing negative selection, preventing those genotypes changing over time, because if they change a bit, those new changes are unfit. And so as a sort of further sanity check, we can ask, well, how many new copy number alterations should we see in our longitudinal series of metastases patients? If we sequence the primary and then we sequence the MET, which is removed from the body, say 2 years later, we've got 2 years of elapsed evolution there. How many new copy number alterations should we expect to see versus how many do we actually see? And if there is stabilizing selection, we should see fewer new alterations than we would expect just if they were steadily accruing over time. So we can make a really simple mathematical model here where we initiate tumor growth at some time 0, mutations accrue over time. Then at some point in the growth of that primary tumor, the met is seeded, that's this TM time. And then later on, we sample the primary, and then at some even later point in time, we sample the metastasis. And so the number of mutations that we expect to be, number of copy number alterations we expect to be divergent between the primary and the met, is the number of copy number alterations accrued along this branch plus those accrued along that branch. And so you can write down just Poisson distributions for what you expect there. You can, from other work from Christina Curtis in Stanford, we expect that metastases seed early, so this time is probably at least 3 years here. So if we, and we also measure in our single cell sequencing what we think the copy number alteration rate is, and it's really high when we look in single cell sequencing, 1 new alteration per 10 divisions. And so if we then look in data, these are the predictions that come out of this simple model, these red lines, and this is the actual divergence that we observe between primary and met. So y-axis here is genetic divergence by different patients here on the x-axis. So you can see that mets are much more similar in terms of their copy numbers to primaries than we would expect by chance. So again, this is evidence of stabilizing selection acting on those copy number alteration genomes. Okay, so going back to these questions then, where does that leave us? So I think if we look at 1 and 2, I think really quite surprisingly, there seems to be a very limited role for somatic mutations in driving metastasis. It seems that metastasis might even be initialized with an optimal genome, at least in terms of the carrier type. So in the last few minutes, I just want to talk briefly about the plasticity angle. What can we say about plasticity? It really looks like if there is adaptation here, there doesn't seem to be lots of interesting evolution going on. So it really must be plastic. Is that what the data tell us? So again, we started doing this work in primary colorectal cancers. We have a study which is called EPIC, funded by the Wellcome Trust, which is a collaboration with Andreas Otteriva here. And in EPIC, what we did was take primary colorectal cancers. We did multi-region sampling from them. And within each of these large multi-region samples, we took a number of much smaller samples. These are individual colorectal glands from the tumors. And we developed an assay to do multiomics on each of these glands. So specifically, we do deep whole genome sequencing, chromatin accessibility by ATAC-seq and RNA sequencing. We have a whole bunch of samples. And so the power of this multiomics is really high. It lets us look at the relationship between evolutionary history that we measure by genomics. We can build a phylogenetic tree from all of the different samples we might have in a tumor and understand the evolution of the tumor. And then we can relate that evolution to the phenotype that we measure by looking at individual glands that we sample, which we primarily do by RNA-seq, but also use the ATAC-seq data. And so we can integrate the analysis here to look at how evolution shapes the phenotype. And what we see remarkably in primary tumors is that there's almost no relationship between genetic history. This is a phylogenetic tree on the left here from many samples from one tumor versus the expression phenotype, which is this heat map here on the right for a bunch of different hallmark pathways that I appreciate you can't read. But the key point here is that the expression indicated by the colors in this heat map bears very little relationship, if any, to the genetic ancestry. So it's most marked between these samples here, where these samples have a very different expression phenotype. This sample here is quite similar to these ones, but yet they have a very different genetic ancestry. So there's a disconnect between the genetics and the phenotype of these cancer cells, and I think that's indicative of plasticity. I'm running out of time, so I won't talk about it, but you can do some mathematical modeling to look at the evolution of phenotype. And these kinds of patterns where phenotypes, gene expression phenotypes and genetics are unlinked is indicative of plasticity. So just in the final minute, I'll show you what we've been doing in metastasis. So following up on this idea, we've started, it's very early days, but we're starting to work on a cohort of colorectal cancer patients with oligometastatic peritoneal disease. So in this particular patient, they had a primary sigmoid tumor, I think, and then it spread around their peritoneal cavity in the location shown here. For each of the tumor regions, we slice it into a number of small pieces, and then we do matched whole genome sequencing and RNA sequencing on each piece. So again, we can build a phylogenetic tree. Interestingly, if we look at the tree, here the color represents different locations around the body. There's really no relationship between the genetic ancestry and the location in the body. So that teaches us that all of the metastases here are highly polyclonal and there's some very complicated seeding pattern going on. Then if we overlay the expression phenotype on top of this, so here the color is no longer location, but is an expression phenotype. So we cluster the most variable expressed genes and overlay those on our phylogenetic trees. You can see that the cells, the tumor regions with the most similar gene expression phenotype are not the ones that have the most closely related genetic ancestry. The colors don't cluster on this tree. Gene expression is not related to genetic ancestry in this metastasis patient. So I think this again is indicative of plasticity. We're not seeing clonal selection here of a particular phenotype. We're seeing the phenotype changing all the time, irrespective of the underlying clonal evolution that's going on. There isn't clonal selection. It looks like plasticity here. So I'll stop there. So I don't think there's any surprisingly good evidence for genetic evolution in driving colorectal metastasis. We see driving mutations are invariably clonal, both in primary tumors in our data and in metastases in lovely data from Johannes Reiter. I've shown you some evidence that I think copy numbers are under stabilizing selection. They don't change within metastases and between primary tumors and metastases. And indeed, Christina Curtis has some lovely work showing that metastases tend to happen early in the colon. So there isn't lots of evolution in primary tumors that later leads to mets. So I think this points to a key role for plasticity in driving metastasis and colorectal cancer. And perhaps if we're able to measure plastic phenotypes in primary tumors, we might be able to predict the occurrence of metastasis from them ahead of time, and indeed perhaps change the way we treat tumors. And just as a sort of concluding thought, I think everything I've showed you tries to dig into the dynamics of tumor evolution in the colon, but typically the only data that we have is from a snapshot in time, a single point in time. And so our ability to get dynamics comes from being able to use mathematical models to ask what dynamical process can produce this static pattern that we observe in our single time point data. So mathematical models, I think, are a way into getting temporal dynamics from single time point data. So a number of people have contributed to it. The main contributors are highlighted in bold, and particularly grateful to Andrea Totariva, who we've collaborated extensively for a number of years. And primarily this work's funded by Cancer Research UK. So thank you very much.